You good to go? Great.
So Manu asked me just this morning to show you the Planktoscope.
Also wear this microphone, sorry.
Oh yeah, no worries. Yeah, so I've been told that he hasn't really introduced this device to you, so I will try to do my best with that. Once I get this flipped in, is that good? Do you know? Oh.
Can people hear Ethan on Zoom okay? Is the sound okay? If you say something.
Testing.
Yeah, I think it's okay.
Yeah, the sound is okay.
All right. Yeah. So the Planktoscope is a device that was developed in our lab several years ago, and it's now kind of what I'm using for my own project as a grad student in Manu's lab. And so this was kind of born out of a citizen science project started in France, which wanted sailors who were just normally sailing around the world to also have a way to sample the plankton along their paths and collect samples for analysis later about like biodiversity and the compositions of the communities and so on. So it was really a environmental monitoring project. And initially they were just kind of having these sailors collect the water, process the water, send the data, their raw samples off to some other place for processing, and the sailors would never see anything of what they collected. And so Manu was thinking, oh, wouldn't it be great if these citizen science project participants had a way to kind of get feedback about what they were doing if they got some kind of visibility into their role in the project. So I think him and another grad student in the lab named Hongchen, who developed some other microscopes in the lab. I don't know if this is something that Manu has talked about yet, the octopi or squid. All right, so yeah, so another grad student in the lab had prototyped a simple microscope to view the samples that the sailors were collecting. Design was intended for the sailors to operate those things. And that kind of evolved with some more work and development into the Planktoscope. So this is really a microscope that was designed for sailors, regular people to operate and to kind of collect data about the biodiversity of plankton communities in the ocean. And then for the sailors to be able to see what they're collecting and send that data off to scientists in marine laboratories for further analysis. So what we have now, don't know what that is. Oh, this is from a different computer. What we have now is basically a $500-ish in parts microscope that is powered by a Raspberry Pi computer, and it has like a web browser interface and so on. But basically, you collect some water, you need to concentrate it to get all the plankton in a smaller volume. And then the microscope kind of pumps water out of your sample, passes it in front of a camera with a lens, saves images of everything, and then that's kind of the raw data that comes out. And then it can also process the data and into kind of isolating the individual objects that pass in front of the camera, which you can use for quantitative analysis or to make pretty collages like this. So yeah, that's kind of the summary of the Planktoscope. And so there's also some downstream processing that we've integrated with to help people label every object that comes out. Say this is a diatom that is a dinoflagellate. This one might cause shellfish poisoning, those kinds of things. And so there is kind of work on getting automated counts of plankton from the data that is collected with the Planktoscope. Yes. And so here's kind of a breakdown of the things inside. And this is kind of what I'll be explaining in more detail when I do a teardown of this unit, which is currently not working. So there's a computer and a board that controls some motors. The motors that are on here, one is this kind of a pair of motors for focusing the image. I'll explain that a bit later. There's some lenses in front of a webcam, basically, that's connected to the computer. And then finally, there's a pump. So I'll talk about these parts in a bit more detail. But that's basically everything that exists. So it's a very simple concept. And this is why the Planktoscope costs like $500 when the existing solutions cost like $20,000 to $100,000 because we made this a lot simpler. It does somewhat less things, but it works pretty well, and the images come out just as nice, and they're in color instead of black and white. So that's kind of the trade-off that we're making, which is a bit of sacrifice on some dimensions, but still really good performance on other dimensions. I think that's kind of how Manu tends to talk about frugal science. So I mentioned there's some data processing with the images that are saved by the Planktoscope, and you can just see this is kind of what happens. We get these crappy webcam images, and then they're corrected a bit, and then we isolate them, and then we upload them to a database. Yeah, so that is the overview. I could talk a bit more in detail about how the Planktoscope works. Do you think that would be?
Let's do a survey if people are interested in going into the mechanics of Planktoscope.
Yeah, can
Raise
we
your
ask a
hand.
question?
Oh, yeah, go ahead.
So I see that you have a GPS on the thing, and I was wondering, why don't you use the phone GPS and just take that out? So it would be even cheaper and more reliable, maybe.
Yeah, the GPS was a early design decision that I think the project is now kind of moving away from anyways. But the reason is that when we collect the data with the Planktoscope, we want some metadata to be associated with it immediately. So stuff like when was the sample collected, when did imaging start, what kinds of processing was done, for example, what was the concentration or dilution factor of the sample, things like that. And it's especially important to have kind of tag of where the sample was collected because plankton communities vary over geography.
Yeah, no, I was just thinking that you could use the phone as an auxiliary, so you don't have to, like you
Yeah.
can just use somehow of the phone to tag those things, what you're asking for.
Right. So the challenge there would be we would need to get the GPS information from the phone onto the
I
Planktoscope.
see.
And so it's probably possible, but I think it would make the user experience a bit more complicated.
I see. Okay.
Yeah. Because right now the graphical user interface of the Planktoscope is just a like web app, single page app that's served from the Raspberry Pi itself. And then so you're rather limited in what you can do with that. Regarding like integration with a phone.
Thanks.
Yeah, it sounds like people are interested in you going into kind of the inner mechanism. Maybe you can do a light version of it.
Sounds good. Yeah. So I guess the most basic level is the Planktoscope is a microscope. And so I kind of think there are two different kinds of microscopes. One is kind of like a magnifying glass. You just have a lens, and then in order to get the thing you're seeing to look sharp in your eyes, you have to kind of move the lens around. So this is kind of what the Foldscope is. I'm sure Manu has talked about that already.
Actually, not too
He has
much.
not. Okay,
too much about-
interesting.
Yeah.
All right. So the Foldscope is also kind of a very low-cost microscope. I think it's a few dollars. I think this is maybe one of Manu's more famous projects. But the idea is basically you can fold up paper in a way that allows you to kind of do things you might normally do with the microscope, like moving a sample around or changing the focus to make the image sharp. And so it's super low cost because it kind of makes things a lot simpler. And the way it works is you just have kind of this glass bead, and it's positioned very carefully in front of your sample so that when you look at it this close, then you can see like the stem of a plant or whatever kinds of cells or bugs at a high magnification.
And we are also working on getting you all the latest version of the Foldscope. So that will be arriving in the next 2 weeks or so.
Yeah, so that's the Foldscope. It's a simple microscope. Then there are these things called compound microscopes, which basically have two lenses, and the optics work in some way that I personally don't know quite as much about. But here the idea is you might like move your sample closer or farther from the lenses, and that is how you can get the image to look sharp when you look through the microscope. And so this is basically what the Planktoscope is. Basically, we have these two lenses here that I can show you in more detail when I do the teardown. And then we have our samples are here, and then these two motors basically move it closer to or farther from the lenses. So yeah, the Planktoscope is a compound microscope. And it has this pump that moves the water sample through this path in front of the lenses that go to the camera, and it saves those images. So how does the pump work? It's a peristaltic pump. It's this big chunky thing up here. And basically it has these three rollers and this tube here, and it squeezes the tube in order to push water through. So it's kind of like when you're squeezing a tube of toothpaste, you kind of like squeeze from one end and stuff comes out the other end. Here it's kind of doing the same thing. It's squeezing the water out this way and it's sucking the water in that way. So this is a very common tool in, I think, bio labs where they need to move fluids through without having the stuff inside come into contact with mechanical parts. Everything is isolated inside this plastic tubing. So basically the path that the sample takes is we have some raw sample that we want to image, and then it gets sucked through this imaging chamber in front of the camera, and it's sucked by this pump, and it gets pushed out into kind of our tube of stuff that we already looked at. I don't know if this in video will work. But basically, this is kind of a sped up version of what you might see when you're running the Planktoscope. This is what you would see in the camera preview. You just see all these cells flowing past. And images are saved as the cells flow past. And so I had mentioned that the Planktoscope has a computer, which is here. And this is what controls all of our electronics and hardware and anything that has interesting behavior in the Planktoscope. So that's just a Raspberry Pi, which is around like, I think $60 or so. And then the Raspberry Pi makes its own Wi-Fi network, kind of like a Wi-Fi hotspot mode on your phone. And then you have a phone or a computer that connects to that Wi-Fi network. Open up your web browser, type in the URL, and then there is a user interface for doing things. Yeah, and I think the final thing I'll just briefly mention is how do we collect the samples that we look at with the Planktoscope? The original design for the Planktoscope was to use samples collected from these specialized things called plankton nets. These are basically just very long nets with the bottle at the bottom. And so as you pull it through the water, either horizontally or vertically, then the water flows through, but the plankton inside get trapped on the mesh of the net, or they just go all the way down into the bottle. And then that is kind of a very concentrated volume of your water sample that has mostly plankton inside, or microplastics or whatever other trash that people have put in the ocean. So that basically, the water from that gets put into the Planktoscope. So I think that is kind of the full view at a high level of the Planktoscope, or what it does, how it works. Any questions so far?
Talk a little bit about the Arctic trip and what you did with the Planktoscope there.
Yeah, so over the summer, Manu had asked me to be a last-minute replacement for someone else in our lab to go on this 45-day trip through the Arctic. Let me see if I can show on the map where this is. So this was a trip that was led by a lab in the Stanford Earth Sciences School. They study the Arctic sea ice and the plankton that live there. They, like in 2014 or something around that time, they had found in the summer under the sea ice, there are these giant blooms of plankton. So the plankton just grow a ton. And then those plankton will eventually die and or get trapped in the ice for the next year. But then some of the plankton will, I guess, sink to the bottom. And that is what feeds everything in that part of the ocean, from like all the sea stars on the bottom of the seafloor up to the walruses and the polar bears who eat the stuff that's living in the sea that depends on the plankton. So basically that lab wants to understand better what happens to all the carbon that gets sucked up by the plankton living in that part of the ocean, and where does that carbon go? So our lab joined that trip because we have some tools that can help to probe that question. And we also brought some planktoscopes, one planktoscope along, just to help get a way to measure with each water sample how much of the plankton is this type versus that type, those kinds of questions. So that is what we did. We basically went around and around in a trapezoid for those 45 days, sampling the same places for water, and then also doing some work on the ice that was melting over the course of the trip.
Do you have photos of the boat that you were on?
I do. Let me see if I can find this. What would this be? I'm not sure actually if I have an easy way to.
That's okay.
Okay.
Yeah. Were you gonna do a disassembly of the
Yes.
planktoscope?
Or that's what Manu asked me to do. So yeah,
Yeah,
I'm ready to do
So
that.
I'm wondering if people in the back are able to see this front table, or if you all can.
I think it'd be better if we maybe group around.
Yes. Get closer. Yeah.
So this is the first version of the Planktoscope hardware that was released several years ago. There's a new version that's much nicer, but I don't have those. Manu took them all, and I don't know where they went. So we'll just be opening up this one. It's sounds good. Pause share. Maybe I'll just leave. I'll leave the meeting. Yeah. So there's basically this top lid. Because this was the initial design, there are some issues with how things were laid out, but I don't think that is too important right now. But it's kind of, it takes a lot of disassembly to access certain things in the Planktoscope. For example, the SD card that has all the software with that the Planktoscope is running. To make it what?
Is it an intentional design for us to make it difficult to access?
So this was an accidental choice, kind of like how there was this other plastic plate back here with holes for the USB ports, but those were too small. So that's why I removed that back plate. So these were design flaws that were fixed in a later version of the hardware. Yeah, so we took off the top lid. You can see there's some wires that run along here from this circuit board over here to the pump over here. So these are basically the wires that run this motor. So this is what we call a stepper motor. It's a bit different than the most common motors where you just have two wires that you like connect them to a battery and then the motor runs. With this one, it's kind of made for precise movement. So basically it moves one step at a time, and each step is a specific like angle increment. And then so you kind of have to send the electrical signals, the power, in a specific pattern in order to get it to move in one direction or the other. So that takes a bit more work to kind of manage. And so in this version of the Planktoscope, it's handled by this separate board, which I'll show soon. Actually, maybe I'll show that now. This here is a ribbon cable. So this is a cable that connects the camera to the Raspberry Pi. So I'll just remove that. This is another ribbon cable. This one connects the Raspberry Pi computer to the motor control board. And normally there would also be a GPS board on top of here that's stacked, but I don't need it for my work, so I just built the Planktoscope without it. So this is the motor control board. You see these two thick wires here, red and black. Those are the power wires for powering the motors, because these motors take a lot of power. we can't just power it from the Raspberry Pi computer. So instead, we have the separate power input for the motors. So this is the motor control board. There are these things here called screw terminal blocks. This is basically an easy way to connect wires to a circuit board, especially when those wires need to pass a lot of power through them. I think I will not try to disassemble that. I'll just open up some more things here. The board I'm taking out here, this black board with the fan on it, this is just to keep the Raspberry Pi cool. Because the Raspberry Pi, when we do the data processing of all the raw images to get isolated objects from the raw images, it runs the CPU at a high power and it gets warm. So we need to keep it cool so that it can keep running at a high rate for longer. Yes. Okay. What should we do next? I think maybe next we can even tear down the sides. Let's do that. Usually this is where I stop, or I'll remove the Raspberry Pi if I need to access the SD card to like change the software on it. Maybe I'll just do that now. This is another thing where I think the design was improved in a subsequent version. It's kind of hard to access the things that keep everything in place. So this is the Raspberry Pi computer. Here's the SD card that goes in there. So in the new version of the Planktoscope, the Raspberry Pi is laid out this way, and then there's a hole in the back for accessing the SD card. One of those design things that was only discovered after this initial hardware design, which is often how hardware goes. You make a version, you find some issues, and then you have to fix them after you get feedback from users. All right, so next I'll remove these. This is also kind of another thing that's annoying about this hardware version is you have to
May I ask a question? Is it Raspberry Pi 4 or 3B?
Yeah,
Which model
so
is it?
it's possible to run the hardware control stuff and the image acquisition stuff, so saving the images. It's possible to run that on the Raspberry Pi 3B. However, I think all the image processing stuff will run out of memory if you don't run it on a 4 with at least 4 GB of memory. So yeah, it's really only designed right now to run on the Raspberry Pi 4.
Another question in the chat,
Okay, I get it. Thank you.
what you use for object detection?
Yeah, so what we have for object detection right now is basically a custom program that uses OpenCV to do some image cleanup, and then another library called scikit-image, which you might have heard of if you use Python for image processing stuff before. And we just call some function there that picks out all of the objects for us. But there are other people in the lab who are working on trying to make better and or more efficient object detection using machine learning stuff, which is not something I'm working on. So, okay. So this is now those 2 sidewalls removed. This is going to be annoying. I'm just going to leave it like that. So now we're getting to the core of the Planktoscope. There's this motor here, which I'll just leave around. And then there's this stuff here, which has the camera for the Raspberry Pi and the 2 lenses and this sample chamber holder. So this device was designed to be kind of modular so that you could like remove and swap out different lenses to get different levels of magnification. Although then you have to change some other stuff, like the calibration of how large is a pixel. Is it 1 micron of the real world, or is it less or more? Those kinds of things. And then also image quality changes depending on the lenses that you use. For example, it might be harder to get things in focus because maybe the range of things that will be in focus gets smaller. So there are all these other things that the project learned as it went. Yeah, this is also one of those tricky things about the Planktoscope hardware is how to assemble this specific thing because it has to be done in a certain sequence. I don't know if it's really worth doing that here. Yeah, I don't know. This is kind of everything I had in mind. I don't know. Does anyone want to see any other specific part of this Planktoscope?
Can you explain again what the GPS thing was, the other one?
This thing? Yeah, so this blue board is the motor control board. So it's able to control either 2 stepper motors or 4 of these regular DC motors. We actually have 3 motors in this thing, but 2 of them can be controlled in the same way. So basically, we use one of these stepper motor outputs on this board to control the pump, and then the other one controls both of these motors together in the same with the same signals, basically. Yeah, good. Yeah, I just finished the teardown of this V2.1. Yeah, so.
So Adam is a postdoc in the lab who also works with Planktoscope for kind of a different aspect of it. And so Ethan just went through the disassembly of it, and then Adam is going to do a demo of how it works. We also have a student who brought some water samples, so maybe we can use that. And maybe while Adam is getting set up, you can talk a little bit more about the citizen science aspect of it, and specifically what you're working on in terms of creating a network of Planktoscope.
Okay, can I ask a question?
Oh yeah.
Oh yeah, go ahead.
So, does it do any auto focusing? Is that why the motor is there? Or is it like fixed focus and then you just flow and whatever is in the field gets imaged?
Yeah, so right now it's basically adjustable focus, but you have to press some buttons in the software to change the focus. There is a feature request for the software to add an autofocus algorithm, which is definitely doable. We just haven't gotten around to it because it's not such a high priority for us. Usually
So-
the way I use the Planktoscopes is I just focus it once and then I just leave it there for like months or a year or 2 years or whatever. It's not really something you need to touch most of the time.
I see. So what is the motor for then? Like the 2 motors on the side, the steppers?
Yeah. So the 2 motors are just for getting the sample in focus initially, or if you need to adjust the focus because you change the lenses or whatever.
I see.
Yeah.
Okay. Thanks.
Yeah, so my project with the Planktoscope is basically I and some other people that were helping out on the project built this rack with 8 Planktoscopes in them. One of them broke. This is the 8th one that broke. And so basically we have them all connected up to the internet, and I just reconfigured the software so that it saves images directly to the cloud, and then we can run our processing after the fact. And we also attach each of these Planktoscopes to an aquarium that we fill with water from Jasper Ridge, which is maybe like a 10-15-minute drive from here. And so we want to do some ecology experiments. So we want to see, like, if we just get the samples into the aquariums, that's kind of a big change for everything living inside. What does that do to the populations? How much random variation is there between these different samples? Do the populations change drastically, or do they all kind of follow the same trajectory? What happens if we add perturbations to these aquariums, like changing the light exposure or acidity or those kinds of things? So we want to kind of develop a proof of concept of using the Planktoscope to do experiments with ecological samples to understand the complexity of the ecosystems around us.
I guess have you like thought about like just having water from different, I guess, ecosystems and then see what happens with it?
Yeah, so actually this was what we did in the first test of the Planktoscope system that I built back in September. We went to Jasper Ridge, we collected some water from the lake, and then we collected some water from the bottom of the dam below the lake. It's totally different, but we haven't looked in detail at that data yet. Yeah.
Are there any questions, even in general, about ocean monitoring or ecosystem monitoring? Anything you're curious about? That's okay. Maybe we should transition. This is Adam. Can you give yourself a quick intro? You want to hand over the mic?
Oh, yes.
Yeah.
So I'm a postdoc in Manu's lab. I came from like a biological background. So I did grad school at UCSF in like protein biochemistry. So I really didn't have any engineering background, but I joined the same day that Thibaut Polina joined. So he was the one that was working on this. And it looked quite different when he joined. So it was actually like this very modular stack. I don't know if you showed them that. But so there's been several design iterations, which was kind of cool from the open aspect of this. So that model, it was great. So it had multiple stacks and they all went together with magnets. But it took, like probably 8 hours to put together because it was like quite complicated. So when we wanted this to be like an open project, we knew that people with different kind of expertises would be trying to replicate it. So obviously like an 8 hour assembly was not going to be really feasible, as well as you needed a bunch of soldering and then like a lot of stuff that a lot of people weren't probably comfortable with. So then this was probably like the second or third version in which I think like 70 people during the pandemic just downloaded the plans. So you just get basically like a little file that has all these kind of drawn out. And then all you really need is a laser cutter, which again is kind of hard. But surprisingly, a lot of people had access to stuff like that. So then they just downloaded that. And then all this stuff, I'm sure Ethan told you, like this is all just off Amazon. So this was like very easy to get. And for like a few $100, you could get it and then replicate it. And so like about 100 people put them together through the pandemic and started using them, which was great. Um, so now we have this version. I'm sorry, I learned this morning I was doing this. So this is kind of the form factor of the newer version. So it's a little bit more robust. This one's made out of wood, the version that they're actually selling. So Thibaut started a company because a lot of people were like, okay, this is great, but I don't have the time to build it. Like I don't want to build it. I just want to start using it to image plankton samples. So then Thibaut started this company in France called Frugal, and so they actually manufacture these. So you just pay them, a few $1,000s, and then you get a little Pelican case, and it has everything you need to start running with the software and everything kind of on board. So people have been using it for a lot of things, right? Initially, it was all research, but then I think, again, another cool aspect of the open science stuff is people would join the Slack and be like, oh, I'm an oyster farmer, and I really need to know when the larvae are coming out so that I could put in these plates so the oysters land. Like, do you think we could configure this to look at the oyster larvae? And so then we were like, of course, yeah. So we changed a few things. Mostly it's just the flow cell that you need to change. So this is like depending on the thickness, right? That's going to determine the kind of sample you can look at. So for the oyster farmers, the larvae are like quite big. They're like almost a millimeter. So we just made a bigger flow cell. Thibaut went out there and then it was great because like that really helped them, right? Because before they were kind of guessing when they were going to come, like based on timeline. But again, with like the climate change and stuff, the timelines were changing a lot. And if they put these plates in too early, like a bunch of crud will grow on the plates and then the oyster larvae won't land and they'll like lose their whole harvest. So they were like super stoked to see this. And fish farmers, again, they wanted to look at copepods, which are these like little shrimps. And so that's what, if you go to South America or Southeast Asia, there's a lot of fish farms and almost all of them use copepods to feed the fish when they're young. So they really wanted to have a tool that would look at like copepod abundance. And so we made another version that's going to be deployed a lot for that. So it's really cool seeing what people would use it for, not just like academic research, which is great, but like not super impactful, right? But when you can actually help people that are trying to like grow fish for food and stuff like that in these places that can't afford an instrument that costs like $100,000, right? Because like in the paper we published, we took the same sample and ran it through. There's an instrument called FlowCam, which is like $150,000. It's huge. It sits on a big bench top. And we ran through the same samples and you could see like side to side comparisons of from the Planktoscope and from that. And like, I would say ours are better, right? Theirs are like black and white, ours are color. Our camera is better. Like their thing is very great, but it's like not very accessible. So it was cool to see people use this. I think it's nice when you also build it like this, you're not afraid to take it apart, which is like another cool thing of the open aspect of science. So I've seen the flow cam and like when they're broken on these boats, like nobody wants to touch it because it's like 150 Gs, right? Like you do not want to open it up and like ruin the warranty. So with this, like if something breaks, right, you're not so afraid to like put a new lens in, or like if you get water on the board, it's not bad. It's like a $30 board. You can just replace that, swap it in. So it's cool to kind of have that aspect of the open kind of hardware and open software. And Ethan, right, has been working a lot on the open software aspect, which is cool, right? Everything you see is on GitHub. So if you're confused about how something's working, you can go through, you know, ask ChatGPT, like, what is this Python script doing? And then be like, oh, can I add something to that? And that helps a lot, as well as knowing what's going on. Because I think, again, as a scientist, when you use microscopes, another like really frustrating thing is the software is always terrible and you don't know how it's working. So when it breaks, you have to call Nikon, and then it takes them a week to come, and then your experiments are kind of shut down for that. So for this, people have been able to kind of cater it to their needs, and those are all on that GitHub, right? So somebody wanted to do like a time lapse, so they made a little script, and then a lot of other people are like, oh, wow, that's great. So I can go and download that and do that. So unfortunately, did you show the software at all?
No.
Doesn't work.
Raspberry Pi's power connector is flaky.
Okay, yeah.
Moving around.
So I don't know how interesting this is going to be for everybody. Certainly not for the Zoom people. So the neat thing I think I really liked about the Planktoscope is because the Raspberry Pi, it can create its own hotspot, its own internet. So you don't need any other computer with downloaded software to run it, right? So all I have to do is hit that little button. So now that's on the Planktoscope. And then all you have to do is you'll see that Wi-Fi network here, and you just join that. And that's all you really need to start running the Planktoscope. So again, for people that were in Southeast Asia and South America, a lot of them don't have super powerful laptops or laptops at all. So showing them they could just use it with their phone, like they were super stoked on that, because just with a cell phone, right, you could run any of these acquisitions as well as you could do on a laptop. So any tablet, anything that has internet, you can start running these experiments. So that was really kind of neat to see how empowering that was for these people that don't have access to computers and stuff like that. So all you then have to do is enter in this this web address, and then now you're in like the home of the Planktoscope. So you can do all your controls from here. And so this is kind of where you would enter in your sample information. So for the people that are doing like hardcore marine biology, right, it's really important they know where the samples came. So if you're on a boat, you'll put in your GPS of where you like threw the net in that you're trying to capture the plankton, where you took the net out, etc. If you're trying to do it from like depth, we have all those tools in there. And this is always great when you're going back and looking at the information. You have to really know where it came from and why that's important. Again, this is going to be terrible because I don't have the other lens, but this is kind of where you would set up the experiment. So like we said, like these are the really overbuilt focusing mechanisms. So the stage is moving, everything else is kind of stable. So what you would do, if you were looking at something that was very small, right, you would want to focus on that. Something much bigger, like a copepod, you want to focus on that. And in this way, you can set all the exposure settings for the camera. So this is where you would get your images like really nice and good. You would then continue. This just looks like a weird light hole, right? Because I don't have the other lens. So because these are like a conjugated lens pair, it's kind of like if you've ever done macro photography with a like SLR camera, you basically just flip a lens. So one is making the image bigger and the other one is making it smaller, projecting it on the camera sensor. So it's exactly what we're doing here, which is like a nice hack to make basically like a objective without spending like hundreds of dollars on an objective. And these lenses are really high quality. They're made for like manufacturing and like surveillance and stuff. So like they're very high quality lenses, but they're, you know, 10 or 12 bucks each. And by changing the focal length of these different lenses, you can change your like effective magnification. So if you're looking at something big, you would kind of swap this out to a larger focal length distance, and something smaller, you would do the opposite. And this way you basically have where you have like changing objectives on a microscope, like you would like on a turret or a regular microscope. So in this way, you could set up your experiment there and see like, okay, my lens pair is like really nice. I can see everything really well. And then you would go and make your experiment. So then this is where you kind of would like automate your experiment. So you'll tell the pump to pump a certain amount. All your stuff is in focus. And then you'll say take 10,000 images. And this will then pump through your sample. And because, again, we're using like a Raspberry Pi camera, which is great, but it still has what's called like a rolling shutter. So you can't take pictures of things that are moving super fast. So the strategy we do is you just pump, stop, take an image, pump, stop, take an image. It's like a stop flow technique. And so this way you could take your like 10,000 images, and then you'll take through, and then all on the Raspberry Pi, you'll then tell it to go through each image and find any objects, right? So this is nice if you have like a very dilute sample, right? Because a lot of times you'll just have blank images because you'll just be looking at water, right? Say you're looking for like a rare dinoflagellate or something that you know is there, but you don't think it's going to be in super high concentrations. So in this way, it's great with computer vision, right? Because the computer vision doesn't care. It's just going to go through, if it sees an object, it's going to take that, cut it out, and then store it. So that way it's really nice because you don't have to manually go through each image and try and find what you're looking for. And so that's called segmentation. So it'll go through and segment out objects. And then you'll get stuff like this, where you'll get all these different objects, and these will be like all the different creatures that were in the image acquisition. Yeah, I could do that. No, it's fine. Yeah, we can go through. And so this is like, I'm sure everybody's heard about like the whole AI image processing that's happening. This is like the most boneheaded image processing you can do, which is it'll go through anything that's like in the previous image, it'll subtract. So you'll subtract the background, and then just doing a thresholding. So anything that's like not blank, it's going to find and cut out. And that's where you get all these different objects. So it's agnostic to anything that's there, but it'll find that and cut it out. And then you'll get these kind of nice vignettes. Did you send it to me? Cool. Oh, I don't have internet, so that'll be interesting. That's okay. I mean, I'll just go through the. Here we go.
Recording in progress.
Got it. Screen share. Okay, everybody can see here, right? Oh, great. Yeah, so this is the whole software architecture, right? So you'll have your cell phone or your computer. What's nice is this is creating the hotspots, so you don't need anything else. You'll do your acquisition. You'll tell the Raspberry Pi, okay, segment, find all the objects. And this is the important thing for people that are doing quantitative science. So Ecotaxa is a website that's operated by a French group of marine biologists. And what's nice is you'll upload all these little guys, and if you don't know what they are, there'll be what's called like a random forest classification, which is another like boneheaded AI, and it'll take a first pass. It'll be like, okay, I think this is a copepod, I think this is a dinoflagellate. And then what happens is actual marine biologists will go through and validate that. So they'll go through and like, absolutely, this is that weird copepod, this is a dinoflagellate, this is a diatom. And in that way, if you're a citizen scientist, you can be super confident that what you took is like actually like bona fide good data. And then what's nice is because this is all like pretty open, then any other scientist or citizen scientist can go through and be like, oh, okay, in this little watershed in Croatia, there are like tons of these dinoflagellates. I can go there and study them. Or, oh no, there's some kind of harmful algae that's there, so we should start thinking about that. Or this is a good place to like do the research. Yeah, this is again how the image processing works. And this is something that we're really excited to kind of push. So as the compute stuff gets better, like as the Raspberry Pis get better, hopefully we'll be able to do more kind of sophisticated image processing, right? So what you can do now on like slightly better computers is you could train the computer to actually find something that you want. So say you're into some toxic algae, which a lot of these people that are living in places where they're fishing for sustenance, often they're kind of devastated by like certain types of algae, and they cause there's many sicknesses, but the big one is this paralytic shellfish toxin, which is like really terrible. So as you start eating shrimp or any of these things that kind of accumulate it, you'll start feeling kind of sick, and then as it progresses, you'll just get sicker and sicker and sicker. And it's kind of like weird symptoms initially, but then as it progresses, you actually get like really terrible neurological disorders, which is hard to kind of diagnose from the sickness, but what's very easy is it's one species of dinoflagellate, and it's very easy to see. So what we are kind of hoping is you could get these into the hands of people that are actually consuming these fish, and if they see that, they can say, you know, stop fishing for a few weeks while it's a problem, and then wait, or just warn people that are going to be eating this fish. So, but what you can do now, right, which I've done in the lab, which is you can train. So I study these algae, right? And they look very different depending on where they are in their life stage. So what I did is I trained a computer program to look for these different states. And then you can train something that will go through and say, okay, with 99% confidence, this is like this vegetative state. So you can imagine you could do this with, say, toxic algae. And then I am certainly no like software expert, but I was able to make this where it'll go through and just automatically go through 10,000 images and count exactly how many of each algae are in there. So for this, it'll be very important for people that want to look at, say, toxic algae or something that is like a real problem. And so this was easy for me to do, and I think it's nice because you could then, I put this all up on my GitHub, so people that want to, say, retrain it for whatever they're looking at, they can then do that without being too intimidated by that. Yeah, so this kind of shows how big a problem it is. So it's a problem here, but it's a problem much more in countries that don't have like a lot of good fishing practices, right? And it's because these people are literally fishing to eat, so they don't have the luxury of like waiting or like feeding them in a correct way or doing like everything as like we can do in places where we can afford to, you know, like go and buy fish at the market. So it's a huge problem when you go down to South America and Southeast Asia. And the other big thing is when these blooms happen, it'll kill all the salmon. So in Chile, they have these like beautiful fjords, and it's all just filled with salmon farms. And they're dumping in a ton of fish food, basically. And what that does, it like spikes nitrate concentrations in the water, and then all these algae grow, and then it kills all the fish. And in Chile in 2016, it was like $800 million they lost because of all the fish died. So that's like most of the economy down there, and it was basically all devastated. So if you could have these surveillance mechanisms throughout, you know, for a few $1,000s, have these, we think it could really help these people, you know, stop these things from happening before they are a huge problem. And so this is where I actually brought it to one of these fishing communities. This is in Indonesia. And so these two people had like never used a microscope before. And so we trained them, and this is on like a little fishing boat. And this is what we were able to see. And a lot of these are cyanobacteria, so these guys. And these in high concentrations can be really cause you get quite sick if you drink the water or eat something that's kind of accumulated. So it's great to see that we were able to see, and these people are now using these on like a weekly basis. When they go out on these transects, they do a acquisition, and they're able to look and see if there's anything nasty in the water. What do we got here? Oh yeah, these are the plates I was telling you about. So these are the larvae, and these are the plates. So how it works is you just put these in the water, and then the oyster larvae land, and that's where the actual oysters will kind of grow. And if you put these in too early, then they get fouled with other gross stuff that's going to grow on it, and then the oyster larvae won't land. So we were able to go and like really quickly see the different oyster stages, and now they're going to use these when they harvest these in the spring, and we'll kind of see how that's going. Oh, so this is a cool thing. So I just got back from another company. So this company makes cheap remote control vehicles. So this is like a little boat that you can drive from an Xbox controller. And so they want us, so what we did is we put the Planktoscope on, and so we were able to then take this. And now you can basically drive this offshore. They have a cool setup where they use this zero tier, which is like a VPN service, and a 4G wireless network, which is just shoved in that the little blue part of the boat. And so now from like 3 miles away offshore, you could drive this boat and say go through an algae bloom, take an acquisition, and then come back, and then be able to look at your whatever weird algae you have. And this is the other thing they make, which is like a underwater ROV. So the next stage would probably be to like, if we could get something on and actually go underwater, that would be pretty neat. I don't know what else we want to see. This class is almost over, right? Yeah, I don't know if anybody has any questions on Frugal plankton imaging. Yes.
Hi, yeah. Can you hear me?
So yeah, a lot of times you would want to just basically then go and say like, don't eat the fish that we harvested. And a lot of times these happen just on like weekly timescales, right? So the algae will bloom for like 5 or 6 days and then they'll just crash. So within like just a 2 week timescale, you could really help things because in 2 weeks it won't be a big problem. And then you could say, then start doing it. Or you could say if you were looking and where to put your fish farm, a lot of times they bloom where there's fresh water coming in. So a lot of times, especially in Chile, there's tons of rain. And so normally you get blooms where fresh water hits salt water. And so you could say then move your fish farm, say, 1 kilometer down because there's nothing but water. So you could say find a correct place to do it. So you could basically survey before you were going to set your fish farm. Same thing for the oyster farms or shellfish farms. And as well as what we did, we just literally had it at before they brought their fish to market and say like, okay, maybe we shouldn't do it this week, but like next week, maybe it's fine. I think there's a question.
Yeah, hi. Can you hear me?
Yeah.
Yeah. Like you said that the water comes through the tube for using peristaltic pump, and then there's a chamber where you view the plankton, right? So like, what size do we take? Like, how much is the resolution? How much size can we take?
So with the setup we have now, it's basically for the most abundant phytoplankton and zooplankton. So it's anywhere from like 20 to 300 microns is the size range you can look at. Below 20 microns, you could still see it. So we worked with a group that was counting coral larvae, which are like 15 microns. And for there, you could still see it and count it, but you say won't be able to do like really good taxonomy. Like you won't be able to see the exact species, but say you already know what it looks like, so you'll be able to do some kind of basically, yeah, just like raw abundance. But you could, again, you could change that objective lens and go much smaller. But then again, you're trading off like the field of view as well as the depth of field. So then you're always gonna have a trade-off, right, of resolution as well as like image quality.
So like, what volume of water do we analyze per minute?
What's that?
Like the amount of water, like the volume of water that you analyze per minute that you can use the device to analyze?
Yeah, so each image is basically taking like a few microliters, right? So in that flow cell, you're taking like 100 microliters per image. And then you could acquire, it's acquiring a couple hundred images per minute. So again, this is where like, yeah, there's definitely a bottleneck as far as how much you can pump and take images of.
So do we use this mostly for quantitative analysis or qualitative analysis, or both?
Both, right? So again, with the marine biologists, they're very good at doing back calculations. So what they'll normally do is you'll take a net, you'll pull it through the water at a known rate, and say you'll know that you filtered 200 liters of water. And then you'll concentrate that down to say 50 milliliters of water. And then you'll image that, and then you'll get abundance, say per mil, and then you could back calculate how much that was in the water. So that's what a lot of what the marine biologists are doing. And say you could imagine doing that if you were the fish farmer or something like that.
Because you don't have the second lens, does that mean you cannot do it?
Correct. Yes, that's what that means.
I'm wondering, do a side-by-side comparison of this version of Planktoscope and that one, and you can kind of talk through the design changes that were made. I think that would be helpful for the students here because they're going to be working on projects and they're also going to have to be making prototypes and thinking about what changes to make based off of user feedback and difficulties they run into.
Sure. Again, this is like not the newest version. So I think like the most obvious thing, right, is this is made out of wood. The newest version we have is made out of bamboo. It's actually like bamboo flooring. The reason for that was like we wanted to get more sustainable. I don't know if anybody has worked with like acrylic, but it's really like kind of horrible to laser cut. It smells really awful. It can get really sharp. It's very brittle. It'll break. And so it's just not the best to machine, as well as when you're doing all these little things with the screws. If you torque stuff too much, it'll crack. And then, especially for people, say, that don't have a laser cutter, they were going to somebody and it's like an ungodly amount. It's like $50 just to cut it. And so then if you broke something, you'd be pissed off. I mean, I would be if I downloaded it and it was broken and then you had to start over with another laser cut sheet. So the bamboo is much easier to machine. So this is kind of how we've done it. Again, all the components are basically the same. The one thing that we did do is instead of having all these like neat little hardware attached, that's what these are called for Raspberry Pi, we made a custom circuit board that kind of consolidated a lot of the things. So to control the LED, the steppers, all that, it's now on like one board instead of having all these things to just make it much simpler. And that's kind of what's inside here. But everything else is really kind of the same. It's much easier now to put together. Honestly, what we did is we looked through like a bunch of IKEA furniture to see how they do stuff in a way where you can put it together correctly. I mean, even we use the same literal little Allen key. So that's really all you need now to put this together. And what's nice is it's like a progressive design. So it used to be if you screwed up, you had to take like all this apart. It was super frustrating. Now we went through a lot of these guides and now it's much easier to put together. So what we did is we gave one of these kits to two groups of people that had never put anything like mechanical together or done any like electrical engineering. And they were able to do it in like 2.5 hours from getting the kit to actual being able to take data. So that was really nice for us. Everything is still very similar. So we have like these magnet designs. So these are nice because it keeps things really stable and in focus for when you're doing optics. So if anybody is making something, I really highly recommend using magnets because it's really great instead of using screws. That way it's also easily accessible. I guess the other thing we made, you just have to tap these. So if you guys know what tapping is, it's like where you put in screw holes so you can screw something in. And that was kind of a disaster because again, a lot of people just don't know how to tap things. Acrylic is also not the easiest to tap. And if you tapped it wrong, your lens would be crooked and obviously you're not gonna be able to take good images. So we simplified that by making it just like where you put this in and have like a nut that holds it in. That honestly was one of like the biggest design improvements. But besides that, I think it's quite similar. I don't see any real design differences. The other thing was just, again, making it in this form factor. It's nice because it's very robust. So you can just throw this in your backpack, which is what I do. And then you can just basically bring the power supply and your cell phone or your laptop and be able to take this to the field and run it. And it's super robust. And the bamboo flooring is also great because it's pretty waterproof. So this part is very sealed. So this is where your Raspberry Pi and stuff is. So it's quite robust to when you take it to the field. And if you spill water on it or something, it's normally not an issue. And I've seen these come back like really gross. Like people spill salt water all over them. And because the components are so robust, they're still able to be used when they're in the field. Again, I don't think anything is very different. It's very open, so you can take all this apart and add, say, different things. So some people say they were interested in microplastics, so they wanted to use like a different light instead of this white LED. So you could use different kinds of modalities of light to say, if you're looking at, microplastics coming out into the San Francisco Bay, you could use this by changing a few things, probably changing the lenses as well. So I think, again, the neat thing was the design iterations really all came from the Slack community. So people were having problems putting stuff together or taking things. So we kind of went through, took all those suggestions or complaints, and then kind of built those into the next kind of design. But everything at heart, I think, is the same, which is nice. It's also nice when you're making this and people are actually buying it. So again, we think that it will probably change. Like a new Raspberry Pi just came out, which is like much more powerful. It has a bunch of cool new add-ons. And so Ethan has been working to kind of see at some point when we can kind of implement that in this. But what's nice, then people don't have to buy a whole new Planktoscope, right? They could just buy this new Raspberry Pi, plug it in, download Ethan's new software, put that on, and then run it. And again, we're also trying to do a lot more. I think with software is really what's going to improve. Again, Ethan's working on more stuff so people don't have to take the SD card out. They could just go on, download like you would an update for your computer, and then have all those software improvements just on there without having to open anything up, which people, again, are also slightly hesitant to do.
Magnets and seawater if you see them rusting over time.
Yeah, definitely. But magnets work when they're rusty, so it's fine. Yeah, again, like anything that you put in salt water, I think is something I also didn't appreciate is it'll get really disgusting after a few days. So that's something you also have to try things out before you put them on a large scale because Yeah, anything in salt water is going to, especially electronics, it'll be completely destroyed. So that was another nice thing about making this a little bit more sealed. So this can get splashed and it's totally fine. Whereas this one, I've definitely ruined them by just getting a splash of salt water on these boards and then it's completely fried. Again, it was also nice because this costs like 15 bucks, so you can buy a new one and then you could plug everything back in and be good to go. The bamboo flooring. Well, it's like got like a 30-year guarantee for the bamboo flooring. So it's
80% bamboo, 20% glue or something.
yeah. So again, it's flooring. So I'm sure again, it's sustainable. Like the company is sustainable flooring. So they're using like recycled bamboo. So it's basically like particle board with bamboo. But yes, they definitely are still putting in some kind of solvent, I think, to make it. So it's not like completely beautiful and green, but it's much better than using plastics for everything.
Is anyone like interested in developing like automated and like durable one, kind of like Argo Flow like kind of things, but like it comes in monitoring?
Yeah, so that's what we did with that boat I showed you. So that company is very good at doing automation. So that's a neat story. Why we like them is like 10 years ago, he ruined a surfboard. And then he was like an engineer and he's like, oh, wouldn't it be cool if I could send this surfboard to Hawaii automated and like without any, just set it to go. And so he got pretty far. I didn't make it all the way. But now that company in 10 years, they make that boat, that blue boat. And it's, again, they have the kind of the same philosophy as we have is you have ROVs, right? You can buy, but they cost, you know, $50,000 to $100,000, and they sell their ROVs for like 5 to 6K. So the idea is they basically undercut the current like ecosystem, but with very high quality. So they use similar components instead of making all crazy custom things. They're using a Raspberry Pi. They made custom hats, and so that's what the dream is for us. And we're like now well on our way is with that 4G capability. So basically anywhere you have network connectivity, which is like with 4G, you can get it pretty much anywhere. So what you can do is set their boat to basically do like a Lagrangian sample. So go through and sample. And then you could basically either set the Planktoscope to just take images every hour, like 10,000 images every hour, or you could log into it through that 0-tier network and then look and see if everything is good and say, oh, there's a lot of stuff right here. Let's run an acquisition. So that's like the dream is to do the automatic monitoring. With the same kind of philosophy, what we want to do is just put buoys. So like where you would have a buoy out in the San Francisco Bay. Because I don't know if you guys followed, like the last 3 years, there's been really horrible plankton blooms in San Francisco Bay. So like 2 years ago, it killed a bunch of sturgeon, which is like a big problem because sturgeon are like not all that abundant. And nobody really know why it never happened before or why it's happening. So you can imagine if you have these boats going around or if you had buoys, then you can have a much better idea. Because again, the big thing holding back plankton imaging is really just cost and personnel. Like literally it's a grad student that has to go out. The biggest one I think is in Monterey Bay. So it's 2 graduate students from Santa Cruz and they have to go out every morning. They go to the pier, they take samples, they go back to the lab and they look with a regular microscope and do counts. But you can imagine with something like this, it's just much easier because you have your computer vision that's running. It can go through and like do all the work for you. And instead of actually having to go out, you could just have these. So I think automation is going to be like the next iteration of this. And I think we're almost there, which is really great.
I'm wondering for both Ethan and Adam, either looking back or in the future, what is one of the most exciting applications of Planktoscope for you?
Ethan, what do you think? You got like the
I'm most excited about all the autonomous stuff. It's going to take a lot of work, but I think that's where it'll become really useful more broadly beyond just marine biology researchers.
yeah. Yeah, I think finding the new applications is great. Like finding it through Slack, which was great. People, what they're using it for. And again, seeing this other company, I just got back yesterday from Long Beach, and like they found all kinds of cool stuff where they're using their little ROV to like find dead fish in these fish farms. Like they can go and scoop them up and take them out. And it helped them a lot, right? Because that would totally screw them up, where they'd have to have a diver go in. So finding these like frugal hacks to these things that weren't available before and helping people out that wouldn't be able to afford it. Because a lot of people, you can't afford like a $200,000 microscope. It just doesn't make any sense unless, you know, you're an academic lab that has grant money to spend. But a lot of people are really willing to spend like a few $1,000s. And I think it's been great to see like the community grow and people be happy. Again, because it's like this open community, even if Thibaut or I or Ethan can't answer questions, there's all these people on the Slack. So basically go and people are like, oh, did you ever have it shut down on startup? Like, has anybody ended? Oh yeah, it's the Python service, just restart it, blah, blah, blah. So this community kind of sustains itself and it's been fun interacting with those. So like with a project like that, I think it's been great. As compared to, I think, a lot of the projects I had in grad school, which are like, often quite lonely, right? You're in the lab, you're doing your own thing. Whereas this, it's like a lot of people you get to interact with on a daily basis, and it kind of expands the reach. And if you could also help like a problem that's really becoming more tragic, right? I think with the oceans, like we just don't really know what's happening with the plankton and how that's going to affect the fish and how that's going to eventually affect us. So having like a tool that's going to really help, I think, in that aspect is really nice. It kind of is like an extra motivation for you to like kind of succeed with your thing while also having like a good time, which is great. I think that's a lot of what you've probably seen with these Frugal projects in Manu's lab, right? Like it really opens these up to people that probably don't have access to like the other kind of scientific tools that are like kind of kept in universities like this. So it kind of brings it out to the community.
Are there any last questions for Ethan or Adam? What was the name of the French company?
Periscope. So if you go to Planktoscope, you should all be linked through that. And I don't know, I think this is every week, the class. Yeah, so next week, I'm sure Manu will bring like the newest version. And then, I mean, in 10 minutes, if anybody's interested, we could run through like a plankton sample. I could even bring a plankton sample from San Francisco Bay. And that'll be probably easier for everybody to see it actually running. What's great is it only takes like 10 minutes to run, and then everybody can see how it's actually going to work. And if you want to play with the data, that's also neat.
Shall we thank Ethan and Adam? So, thank you so much. You can that one of the participants in the class, Nia, has built an espresso scope. So I think it would be cool if they could show. Are you still? Hello.
Hello.
Do you mind showing everybody the
Nope.
scope that you built?
Yeah, so this is our espresso scope. It was actually my supervisor that put this together, and we didn't have the magnets, so we are using compression springs instead.
Cool.
Yeah, I don't know. Should I talk through?
You want this?
Sure. What was the intended use case for it?
Just our lab, we like building low-cost microscopes, and we saw this and we're like, this is as low cost as you can get. So we just, out of interest,
Nice.
wanted to see it.
Yeah. Ethan, do you want to say a few words about EspressoScope and why you thought of the idea and application?
Sure. So this is not my project. I just gave some advice on the software and I helped write the paper. That's about my level of involvement in this. But I was working with the person who developed this, Benedict, and the initial motivation for developing this microscope was we wanted to have a microscope that we could just put underwater and watch like stuff that fouls and grows on surfaces, because there's like lots of interesting things that live on the surfaces underwater. That's probably where, in my opinion, the most interesting things tend to be. So the idea is kind of a microscope that you can deploy in the water, get some recording of what is going on underwater in these surfaces. And so basically, Benedict had the idea to use this little ESP32 microcontroller board with a camera module on it. And that is basically got developed into this more general platform with some software that could be easily adapted to different use cases related to microscopy or even like non-microscopy related imaging stuff. Yeah.
Thank you so much for sharing.
So how much did it cost to put that together, Niam?
I think the ESP board, it's maybe something like €6 or something. Well, this is euros for us.
Yeah.
The screws we had in the lab, but they weren't very expensive, and we tried to get the magnets. You can get them on AliExpress. I tried to order them on AliExpress, but they never came. so I got scammed or something, I don't know. But instead of the magnets, we just have a screw and a compression spring. Again, we just had them in the lab, and everything else is 3D printed. The lens to make the actual microscope, it comes with the ESP board, and you just unscrew it to make the microscope. And then by, tightening or loosening the screws, you can focus it, and then you 3D print this little periscope, which you can either spray it, but we just put tin foil on the inside to reflect the light, because the ESP board has its own little light on it, and then you reflect the light onto your sample. So it's great. And then you just connect to it, kind of like the Planktoscope with the little thing on your phone. It's not working for me right now, but so I don't know. I think the main expense was just the ESP board, which was maybe €6 or something like that. So it's like as cheap as you could possibly make it, if you have a 3D printer.
But you followed the prescription as is, like you didn't do anything new otherwise.
The only thing we didn't do was the magnets. So there's
I
a
see.
little hole in there in the 3D printed part for the magnets. And I as far as I am aware, it's you put the same poles of the magnets towards each other, and that makes the that's how you focus it. That opposes two parts, and then you screw them together. Is that how it works?
I'm not sure, because this was stuff that Benedict was doing on his own after he went back to Germany.
I think that might be how it worked, but we just didn't
I
have
see.
them. That's the only change we made.
Okay.
It might also be the magnets might be used for holding a slide onto the thing. I think in the paper we were describing a spring-loaded mechanism for the focusing.
Yeah, okay. Maybe that's it too. I don't know.
Probably really don't need
I
the
never
magnet.
got my magnets.
Thank you.
Thank you so much for sharing. I'm going to remove Spotlight now. And so I think we are going to end class a little bit early today. Thank you again, Ethan and Adam, for coming and talking about Planktoscope. I think that you all can start thinking about if maybe Planktoscope would be useful for one of your projects. All of these open source devices are very modifiable. So say you want to look at microplastics, you can think about how Planktoscope might be good for that, or you may want to build an espresso scope to have a more accessible microscope for low resource settings. There are a few announcements we need to make about deadlines. Do you want to talk about it, Rucha?
Sure, I remember the deadlines. I think by Tuesday before class, we would love for all of you to have either sent your own message or have a thoughtful reply to someone else in the project board discussion regarding topics and things you're interested in. This is the time to start forming groups. So we're hoping that you'll find groups of people that are equally excited about the same problems as you. So please use the Discord community to do that and start making those connections and think about what might be interesting topics and angles for your project. We're hoping the teams will be like around 3-4 people-ish each. But if you find that you're having, a lot of people interested in the same topic, maybe you can take slightly different approaches, things like that. We can work out those logistics later. But for now, get to discussing and have one post. And then I believe there's some readings that were sent out that'd be fantastic for you to take a peek at.
The Fred Park table.
Oh, yes. Is that also Tuesday?
Tuesday, yeah.
Yes, there's also a Discord channel for the Fred Park table, which you can start working on as you talk to people in your group. Remember that it's very useful to have by the numbers done before that. So if you're planning on changing your topic from whatever you did by the numbers on, it might be a useful exercise to do a redo of by the numbers for your new topic. It'll just make your life a lot easier when you get to Fred Park. And keep an eye on Discord. I know we send a lot of announcements, but hopefully it's nice to have everything in one place.
Yeah, and just a quick note about forming teams. I know that with particularly for the in-person cohort, you all may be very familiar with each other and want to work with each other, which is fine, but you also have all the external participants. I would highly recommend reaching out to a few of them and getting to know them as well. I think they can be a very valuable resource, and there's very rarely, if any, other classes at all on campus where you'll get this opportunity to work with someone all all the way across the world. So please try to form teams that are not just people you're comfortable with, but also other people who have different experiences from you. All right, with that, I think we can end class. Thank you all. Can we give another round of applause to Ethan, Adam? Thank you. All right, and we will see you next week. Thank you, Niam, for sharing.